growth of a plant. What a remarkable process it is. The resulting bulk or biomass provides all our food and indirectly nearly all our energy. The process by which plants increase their bulk is called photosynthesis, and it's a process whereby the energy from light, from the sun, is converted into stored chemical energy. It's a photochemical process. And in this program, we're going to see how nature does it. After all, nature has had 3 billion years to learn. And then in the next program, we will see whether we can't improve a little on nature. But first, we must ask what it is that the plant makes and where it gets its materials from. The plant is mainly made of carbohydrate, which contains the elements carbon, hydrogen, and oxygen in the ratio CH2O. Where does this come from? Well, clearly it can't come from the soil, because if you plant a tiny seed or a seedling, it will grow into a great plant which weighs more than the whole of the soil and the pot. Where does it come from? Well, the H2O part presents no problem, because we all know that plants need lots and lots of water. What about the carbon? The plant seems to appear from thin air, and that's the clue to where the carbon comes from. It does, in fact, come from the carbon dioxide of the air, of course. And we can show that by this experiment. If we take some barley seedlings like this, and one of the leaves of the seedling is exposed to radioactive carbon, carbon-14, in the form of carbon dioxide in the air, and then the seedling is placed onto a photographic film, we shall find that the film is darkened. It takes a photograph of itself, an autoradiograph, where it has been exposed to the carbon dioxide, the radioactive carbon dioxide, and also some of it gets down into the root, but not the leaf which has been only in normal carbon dioxide. This is an exposure of only 15 seconds, so it tells us that even in this very short time, the carbon dioxide of the air gets into the plant and, of course, incorporates itself into the bulk as carbohydrate. Fortunately for animal life, there's another product as well as carbohydrate. The little bubbles of gas coming from the leaves of this Canadian pondweed are oxygen. In the light, plants evolve oxygen, so much so that all the oxygen in the atmosphere is made by plants. So, to summarize, plants take carbon dioxide from the atmosphere, water from the soil, and in the presence of light, they make plant tissue, largely carbohydrate, and oxygen. Energetically, this is, of course, a highly unlikely process. When carbohydrate, sugar for instance, burns, the reaction releases 500 kilojoules per mole of carbon dioxide produced. So that's the amount of energy which must be provided to drive the reaction the other way. Somehow light has to provide this energy, but how? Well, it must be absorbed, first of all, and that's why plants are colored. Most of them, familiarly, green. Not only the large plants, but tiny green algae like chlorella have these pigments which make them green. But not all plants, not all photosynthesizing organisms are green. We have plants which have all sorts of colored leaves. We have algae, like the porphyridium, which lives rather low down in the sea and therefore has to collect blue light, and so decides to be red so that it can absorb the blue light. We have the familiar brown algae of the seaweeds. And so a wide range of colored pigments appear in these organisms, all of which photosynthesize and all of which evolve oxygen. Well, to extract these pigments is fairly easy. If we want to study the pigments in spinach, We can put the spinach into a blender with some petrol and chop them up. And then the material contains rather a lot of rubbish, a lot of plant debris, which we can filter off, and so get a nice, clear solution, which is nevertheless still a complicated mixture of the pigments which are collecting the light. To separate the pigments, we use a chromatography column. After some time, the mixture of pigments will have been resolved into several distinct bands. Let's take the largest of these bands and look at its absorption properties. Here, Margaret Westby is examining the material using a spectrophotometer. She set the instrument to scan the absorption spectrum from 760 nanometers down to about 300 nanometers. The first major band is in the red region at 680 nanometers. Then there's a region of relatively little absorption in the green-yellow part of the spectrum, and then another absorption band in the blue region with a maximum at about 430 nanometers. This spectrum is in fact that of chlorophyll A, the most important pigment in photosynthesis. But how good is it at picking up the wavelengths emitted by the sun? Well, by comparison with the spectrum of the sun's radiation as it reaches us on the Earth, we can see that the chlorophyll absorbs it very well throughout the red and blue regions and less well in the green. It only really fails in the infrared region, where in any case the photons have less energy, not enough to drive photosynthesis. So chlorophyll is a good molecule for absorbing the sun's energy efficiently. We'd now like to know how chlorophyll is arranged in plants. Here we've put our Canadian pondweed under the microscope, and we can see small green oval particles moving around slowly inside the cells. Biologists call such things organelles. These are chloroplasts, and they contain the chlorophyll. Under the electron microscope, the detail inside a chloroplast is striking. There are many layers of membranes, which in places are folded over and stacked together like piles of coins. The chlorophyll is contained in these membranes. But how is light collected at the molecular level? Well, we do know from many experiments that within the membrane there are structures containing protein and chlorophyll called photosynthetic units. And we know that these collect the light. Schematically, a photosynthetic unit is often represented as a triangle. The upper part of the triangle is the antenna. This collects light, so it's called the light harvesting antenna. Experiments have shown that in green plants it's made up of chlorophyll, about 300 molecules per unit. At the bottom of the triangle are special chlorophyll molecules of lower energy, probably arranged as a dimer. This region is called the trap, or photosynthetic reaction center. Now, if any molecule in the antenna absorbs light, an excited singlet state will be formed. This excitation energy can be transferred from one molecule to another. If the molecules are all energetically identical and the right distance apart, the energy will travel back and forth in a random way until it eventually reaches and excites the trap, which is at the lowest energy. In some plants, there are several different pigments in the antenna. Here, the energy will be transferred successively to those of lower excitation energy and then to the trap. This harvesting is so efficient that over 90% of the photons absorbed by the antenna produce excitation of the reaction center. And to be so efficient, it must occur very rapidly. In fact, as we shall see, it occurs in a few hundred picoseconds, less than a billionth of a second. Now, to study processes as fast as this requires special techniques, but they are of special interest to us because we are photochemists, and these are the primary photochemical events. that occur in photosynthesis. Let's look first at light harvesting events in the microscopic red alga, Porphyridium cruentum. Like many red and brown algae, it lives at depths where much of the sun's radiation in the red region has been filtered out by green algae near the surface. If we look at an electron micrograph of Porphyridium cruentum, we see additional granules on the surface of the membranes. These granules, or phycobilisomes, contain accessory pigments which enable the algae to collect the green and yellow light which, as we've seen, chlorophyll doesn't absorb very well. Here's a model of the red phycobilisome granule sitting on top of the membrane. And it's made up of layers of protein. It's fairly easily removed, and we can do that if we wish, remove it from the membrane and study it separately. It's red because the outer layer here, which is like the outer shell of an onion, contains a pigment called phycoerythrin, which is red. But we can strip off the pigments one by dissolving them away, and we find that there are several of them, in fact 3 in this case, arranged like this, and slightly different color. They're called phycoerythrin, phycocyanin, and the blue one allophycocyanin. All of these pigments fluoresce. As well as having an absorption spectrum, they have a fluorescence spectrum. Chlorophyll, for example, fluoresces very nicely in the red. Of course, in the light harvesting unit, the fluorescence is very weak, because if it were strong, or any fluorescence is wasted energy. But they do fluoresce sufficiently for us to be able to use this method of studying them. And they fluoresce at different wavelengths. So by filters, we can filter out the 4 different fluorescences and see how the energy is moving around within this phycobilisome. But it's a very fast process. And so the excitation that we're going to use has to be a very short pulse of light. The experiment which we're going to do now is to excite with a short 6 picosecond pulse of light this outer pigment, the phycoerythrin. Let's go briefly through the laser components that allow us to do this here at the Royal Institution. We use a solid-state laser. In the blue box is a glass rod doped with the rare earth neodymium. On excitation by a flash lamp, the laser gives out a series of very short pulses by a process called mode locking. But we only want a single pulse, and we achieve this with a device called a Pockels cell, which selects just one of the 6 picoseconds long pulses. The wavelength of this pulse is then halved to 530 nanometers in the next optical component. Conveniently for us, 530 nanometers is short enough to excite the outer pigment of Porphyridium. Now let's see how we do the experiment. Here, Dr. Godfrey Bedard is using a low-powered helium-neon laser to check the alignment of the components. The beam or pulse is sent via mirrors and lenses to the sample Porphyridium cruentum. The resulting fluorescence emission and decay is then detected by a streak camera over a time scale of picoseconds. After aligning the optics with the help of Amanda Winkworth, Godfrey checks the mode locking of the main laser.
Fire.
He looks for an intense burn on the photographic film. The mode locking is good, so all is now ready.
Okay, that's fine. Still experimenting.
Ready to charge.
Ready?
Fire.
An oscilloscope then displays the real-time fluorescence decay signal recorded by the streak camera. So what does this decay time tell us? Well, what we've done is to send a single 6 picosecond pulse of 530 nanometers wavelength through the sample and then monitor the fluorescence of phycoerythrin at 578 nanometers over a few hundred picoseconds. From the decay curve, we can calculate the lifetime, which is 70 picoseconds. But what we can also do is use different emission filters and look at the fluorescence of the other porphyridion pigments. In this way, we've obtained a decay time for phycocyanin of 90 picoseconds, for allophycocyanin of 118 picoseconds, and the fluorescence of these pigments grows in after the exciting pulse. This is direct evidence that each pigment in the phycobilisome is transferring its energy to the next pigment, which has lower excitation energy, and so on to chlorophyll. But what happens next when the excitation has reached the antenna chlorophyll? This was first studied in green algae, where the pigment was mainly chlorophyll, but the results were misleading. With this single pulse streak camera technique, we and a number of laboratories found lifetimes much shorter than had been predicted. And after about 18 months, we all realized what was happening. We were exciting more than one chlorophyll in each antenna unit, and two excited chlorophylls were annihilating each other. Well, to overcome this, we have to use much weaker pulses, and if we use weaker pulses, we lower the sensitivity. However, we can manage to do this, first of all, by using very sensitive detectors, detectors which will detect just one photon. With this type of single photon counting laser apparatus, we could follow the energy transfer in the chlorophyll of green algae more accurately. Here, Tony Roberts is adjusting the main argon ion laser. Its green output looks continuous, but there are about 100 million pulses a second. These are converted by a dye laser to red pulses. Each pulse is only about 3 picoseconds long. These are shown up by the dust in the room, although we do try to keep our laser laboratories clean. We don't want 100 million pulses a second, so a cavity dumper reduces this number. And the pulses are then directed into the sample chamber. Photons of fluorescence are picked up by a photomultiplier, and the photomultiplier output is displayed on an oscilloscope. Each upward jump of a spot represents just a single photon, and the horizontal axis represents the time delay. In this way, a decay profile of the fluorescence builds up as the experiment proceeds. Here's the decay profile for chlorophyll in chloroplasts. It's the sum of two exponential decays, one with a lifetime of around 400 picoseconds, and the other with a lifetime of around 1400 picoseconds. Both decays arise from the light-harvesting chlorophyll molecules, which can transfer their energy to the reaction centers or traps. If the trap is open, the energy is trapped in 400 picoseconds. But if it's closed, the lifetime of the excitation, and hence the fluorescence, is 1400 picoseconds. The fact that there are two distinct lifetimes indicates that the photosynthetic units really are separate entities, like a number of puddles of chlorophyll, rather than a large lake. So we've seen how wonderfully efficient light harvesting is. The plant arranges its leaves to collect the maximum amount of light, and it arranges the molecules, the pigments within those leaves, to transmit that with maximum efficiency to the reaction center. But what then? what happens in the reaction center. To study this, we really need to remove all the antennae, because there are more molecules absorbing there than there are in the reaction center. But this is difficult to do with the green plant, although much progress is being made at the present time. But for the moment, the best we can do is to study a near relative, the photosynthetic bacteria, which have a simpler system and whose reaction center can be isolated fairly simply from the rest of the bacterium. My former student, Maurice Windsor, has studied the reaction centers of photosynthetic bacteria using picosecond flash photolysis and absorption spectroscopy. And what he and his colleagues find is as follows. In the bacterial reaction center, there are four main components: a protein, cytochrome C, bacterial chlorophyll, a substance called bacteriophyophytin, and a quinone ion complex. Now, when the reaction center bacterial chlorophyll is excited by light via the harvesting pigment, it transfers an electron to bacteriophyophytin in the incredibly short time of 3 picoseconds. Within another 200 picoseconds, the electron is transferred to the quinone complex. The oxidized bacterial chlorophyll is then reduced by cytochrome C more slowly, over 1 to 20 microseconds. In bacteria, the ultimate electron donor is a reducing molecule such as hydrogen sulfide. In green plants, it's water. Many of the details of electron transport in green plants are given in the source text. Here, I just want to summarize the overall scheme. Chlorophyll photosensitizes the decomposition of water into oxygen, 2 hydrogen ions, and 2 electrons per molecule. The electrons then reduce carbon dioxide to carbohydrate. If instead we could recombine the hydrogen ions and the electrons directly, we'd have hydrogen gas, and the overall reaction would simply be the dissociation of water into its elements. So we've seen how in the process of photosynthesis, water is split into its components, oxygen and hydrogen, the hydrogen being used to reduce carbon dioxide to carbohydrate. It's a wonderful process. But from our point of view, our purposes, it isn't quite perfect. It's low efficiency, and carbohydrate is not the ideal fuel. It would be better if we could obtain the hydrogen directly, a very useful fuel. That's what the next program is about. The splitting of water in sunlight into hydrogen and oxygen, but now not in the plant, but in the laboratory.